So, dear audience, my name is Kornél Molnár, and in my presentation I will talk about the characterization of allergic skin diseases in experimental animals. But before I dive into the scientific part of my presentation, I would like to talk about a few words about myself and my journey in the National Academy of Scientists Education. So I started my general medicine studies in 2019 at Semmelweis University, and at the beginning of my studies, I decided to join the Students' Scientific Association, and this opportunity came to me when I passed my physiology exam in 2021, and on that summer I joined the lymphatic physiology research group led by Zoltán Jakus. And after half a year, the opportunity to join the NACE program came to me. So in February in 2022, when it became nationwide, I was one of the first members in the Semmelweis University who joined this program. So both the TDK and this program encourage us to participate in many conferences, national, international conferences as well, also the TDK conferences, which, yes, in some point, some view it's a competition, but what's really important is the experience and the people you met and get to know you. On the left side, you can see me with my friends at the latest national, also the OTDK conferences. On the middle photo, you can see me with the poster at the Korányi Frigyes Scientific Forum. It's a forum held by a dormitory by the Semmelweis University students, and it's quite special because students who are not learning at the Semmelweis University can participate as well. And at the end of this conference, there is always a famous scientist who can or who speak about his job or field. In 2024, it was Katalin, and I got the opportunity to take a photo with her and get her book signed as well. So, realizing these opportunities that given by the NACE and the TDK, I started my MD PhD studies in my 5th year. And during my 5th year, I also spent 2.5 months in Helsinki through the Erasmus program, which is an opportunity that I recommend to everyone to live with that, because you not only have the opportunity to learn in a different culture, in a different country, and to see many things, but you will also have the opportunity to meet with a lot of people and make friendships and learn new things about other cultures as well. Yeah, so what I also want to mention that I was Senior Student of the Month in 2024 in February, and thanks to the NACE program, I've been involved in multiple projects, and thanks to that, it's resulted in my first author and co-author publication as well. And yes, the NACE program comes with expectation, but it can give you back a lot of in return. And with this, you can also get easier several scholarships like the ECAP or the National Higher Education Scholarships, and it gives you the opportunity to join as well as other programs like the Kerpel-Froning Development Program. Well, I graduated this summer at the Semmelweis University, and now I'm a PhD student at the Department of Physiology at Semmelweis University. And having said that, that I wanted to talk about myself and my journey in the NACE. So now I would like to jump into the scientific part of my presentation, which is about the characterization of the allergic skin diseases in experimental animals. So the contact dermatitis is a skin inflammation condition which is caused by a substance that is exposed to our skin, and it indicates induce an immune reaction. This inflammatory response is induced by the so-called heptins. These heptins are not immunogenic on their own, but they are become immunogenic when they conjugate to a larger protein in the skin. The clinical symptoms of this disease is red, dry, itchy skin with bumps and blisters, and this also can cause swelling and a burning feeling. The two main types of the contact dermatitis is the irritant contact dermatitis, which is a non-heptene specific inflammatory response, mainly driven by the innate immune response, meaning that you will have symptoms at the first time when you expose to that irritant substance. While the other type is the allergic contact dermatitis, which is a delayed immune response, meaning that here the main player is the adaptive immune response. So here you won't have symptoms necessarily at the first time when you expose to that substance, but later when you repeatedly expose to that substance, you will have these symptoms as well. To study this human disease, we have the contact hypersensitivity mouse model, and thanks to that, we learn a lot about this human disease, but many questions remain unanswered, like how the lymphatic system plays in this disease, or how the different heptanes induce or influence the induced immune response in this disease. So a few words about this contact hypersensitivity mouse model. It is a great model to study this human disease and to learn the pathomechanism of this human disease and to help us to better understand it and to prevent it or treat this human disease. This consists two immunological distinct phases. The first phase of this, the so-called sensitization phase, it is when for the first time is exposed the animals with the substance. This is when an allergen-specific immunity develops against that substance. But it is important to mention that those heptanes that we use to induce this disease can cause irritant responses as well in the animals. So it is also a great model to investigate the human irritant contact dermatitis. So after that, when we repeatedly expose the animals to that substance, that is the elicitation phase, it comes with an inflammatory response with significant tissue damage as well. And as I said, there is different model allergens that we can use to induce this disease, like TNCB, DNFB, or oxazolone, but the cellular immune responses elicited by them and the differences between them remain incompletely understood to this day. The other question of our studies was that how the lymphatic system play in this disease, and a few words about the lymphatic system. Well, when we start to talk about the circulation of our body, we mainly remember of the arteries, the veins, and the capillaries, and we talk a very few words about the lymphatic system, but it is as important as the blood circulation in our body. It has several well-established functions, like the maintenance of interstitial fluids, the transportation of lipids, and the immunosurveillance. However, the role of the lymphatics in the contact hypersensitivity and in the contact dermatitis, it is still not fully known. So during our experiments, we had the following aims. We wanted to investigate the role of lymphatic vessels in different phases of the contact hypersensitivity disease, and we wanted to characterize the cellular immune processes involved in the pathogenesis of contact dermatitis induced by different heptanes, in our cases TNCB and DNFB, after single and repeated exposure. And for that, we had the following methods. During our experiments, we mainly used two types of mouse models. The one is the wild type mice, which is regular mouse with without any genetic modification. The other mouse model was a transgenic mouse model in which we could locally delete the lymphatic vessels in a given specific area with injecting a toxin. And in my presentation, this pinkish area illustrates where we deleted them. So in some cases it was the hind limb of the animals, and in some cases it was the ear of the animals. On this timeline, I would like to show you that how we use this mouse model in the contact hypersensitivity mouse model. As you can see on this timeline, prior to the sensitization, we deleted locally the lymphatic vessels by injecting that toxin. Then on day 0, we treated the animals with the heptane on their belly, and after 5 days, we elicited with the same heptane the animals on their ears where they were lacking lymphatics. So this was that specific special mouse model that we used in the contact hypersensitivity. And after that, to investigate the differences between the heptane, the induced immune response by the different heptanes, and the effect of the single and the repeated exposure. Firstly, for the single heptane exposure, we had animals that were treated on their ears with either with DNFB or with TNCB, and of course we treated the animals with a control molecule, here the so-called vehicle 1 or vehicle 2. Then to investigate the repeated exposure and the effects of it, we used the classical contact hypersensitivity model. Here we treated the belly of the mice with DNFB or with TNCB. TNCB, of course, with the control group as well. And 5 days later, we treated the ears of the animals with DNFB or with TNCB. And also having group where we had a different sensitizing agent and a different eliciting agent, meaning that some of the animals were sensitized with DNFB on their belly, then treated with TNCB on their ear, and of course, animals who were sensitized with TNCB on their belly and elicited with DNFB on their ears. And what we did after all these treatments, in all cases, was that we measured the ear thickness of the animals, which is a great indicator of the inflammatory response induced by these heptanes. And after that, we sacrificed the animals and collected tissue samples for histological samples, and for a method that is so-called flow cytometry analysis. Here we analyzed the cells one by one and then characterized the cell population based on that. So now I would like to show our results. So when we investigated the role of the absence of the lymphatic vessels in the contact hypersensitivity, here you can see the timeline of this experiment as a reminder. So after repeated antigen exposure, we could detect that those animals who were lymphatic deficient in their ears at the repeated antigen exposure showed a significantly larger ear thickness enlargement compared to those animals who got intact lymphatics in their ear skin after repeated antigen exposure. Here on these histological sections, you can see this confirmed by that you can see that this ear is significantly larger, thickened compared to the healthy ear, and what you can also see that there is a way higher number of infiltrating immune cells in the ear. So these results suggested already that the lymphatics has a crucial role in the contact hypersensitivity. Following that, examining how different heptanes can influence the induced immune response, and also the single and the repeated antigen exposure. So here we made experiments with the single antigen exposure and with the different heptanes. And what we could see that both antigen single exposure significantly increased the ear thickness of the animals. So also the TNCB treated group and also the DNFB treated group showed a significantly thickened ear compared to their control groups. And what we also see that the DNFB treated group showed a significantly larger increase in their ear thickness compared to the TNCB treated group. and this was confirmed also by our histological sections. Here you can see that both treated group was significantly thickened compared to its control group, but the DNFB treated group significantly larger compared to the TNCB treated group. Here you can see the results of our flow cytometry analysis, so where we analyzed one by one the cells and characterized the cell population based on that. Our results show that the single DNFB treatment caused a significant, almost significant increase in the neutrophil granulocyte numbers. The neutrophil granulocytes is a type of white blood cells that are among of the first-line defenders of our body, and they circulate with the blood stream, and they can quickly arrive to that site where is the harmful agent or bacteria try to enter our bodies, and they are an important player of our innate immune response. While on the other hand, the TNCB-treated treatment caused a significant increase in the macrophages and the dendritic cells. These cells is also an important type of white blood cells, but they have a different functions. More about the communication, which means that they take up these antigens, these harmful agents, and then present this information to other immune cells, and by this helping activate the adaptive immune response, which is a more specific and long-lasting immune response to a harmful substance. So these results already suggested that the DNFB acts more like an irritant heptane that induce the innate immune response, while on the other hand, TNCB acts like a classical allergen. After that, we investigated the repeated antigen exposure and this effect on the induced immune response. And what we could see that repeated DNFB treatment didn't cause a significant differences in the ear thickness, meaning that it was not important what we used in the sensitizing phase. If we elicited the animals with DNFB, you can see that these ears quite similarly thickened. While on the other hand, repeated TNCB treatment only caused a significant increase in the ear thickness when we sensitized the animal with the same heptane. So if we sensitize them with TNCB as well, and here you can see that this is also confirmed by our histological sections. We also made flow cytometry analyses, so again analyzed the cells one by one and the population based on that. And our results show that those animals who was sensitized with TNCB, then elicited with TNCB, this treatment caused a significant increase in the leukocytes, so in the immune cell numbers, in the neutrophil granulocyte numbers, and the dendritic cell numbers. So and this treatment caused a significant increase in the immune cells and a significant inflammatory response compared to the other groups, other treatments. So based on that, we had the following conclusions. When we investigated the role of the lymphatics in the contact hypersensitivity, based on our results, the lymphatic system plays a crucial but dual distinct role in the two phases of the contact hypersensitivity. And when we investigated the differences between the induced immune response by the different heptanes, we could see that the DNFB induced an irritant processes, while TNCB triggered a classical immunization, and repeated DNFB exposure did not produce a stronger inflammatory response, while repeated TNCB exposure did. So that all indicated that DNFB acts more as an irritant heptane, while TNCB triggers a classical type 4 delayed hypersensitivity reaction. And what we saw also that I didn't show you, that we observed cross-reactivity, so the order of the heptanes applied during the first and the repeated exposure is not negligible. And we hope that our findings may contribute to the selection of the optimal model allergen for studying the different processes occurring in the sensitization and the elicitation phase. And the results of these experiments has been published already, so if you are interested in, you can read it, of course, and you can find me and ask any question in the coffee break. And I would like to thank my supervisors, Zoltán Jakus and Gábor Kovács, and all the members at the lymphatic physiology group. And there is a photo of us, of our laboratory, and thank you for your kind attention.
Thank you. And now we would like to present the certificate.